All right, well, thanks, Tony, for inviting me here. So my name's Adrien Ponticorvo. I'm a postdoc now at the UT Health Science Center. I thought I'd talk today, hopefully somewhat informally, so if anyone wants to interrupt me to ask questions, please feel free. I normally say that at the beginning of talks. It rarely happens, so who knows. Today, like Tony said, I'm going to talk about some of my PhD work that I did with Andy Dunn, developing new optical techniques to look at the brain, and then time permitting, hopefully I can maybe talk briefly about some of my work now, trying to adapt some of that stuff to look at the retina. The three techniques I'll sort of be talking about today, laser speckle contrast imaging, multi-spectral reflectance imaging, and phosphorescence quenching based quasi-imaging. I'm sure a lot of you here are at least somewhat familiar with a lot of these techniques to varying degrees, so I won't spend too much time going over the backgrounds of how they work, maybe try and focus more on some of the applications and some of the research I've done. As opposed to the standard outline, I thought what I would try and do is just eliminate that slide altogether and sort of tell the story chronologically of how and why I sort of became involved with each of these techniques. So like Tony mentioned, this actually started for me out of grad school. Out of undergrad, I actually started working in industry for a few years at IBM, designing high-end computers. On the left there, you see the deep blue machine that beat Kasparov in the late 90s, and to the right, the more recent phenomenon of Watson on Jeopardy. I did not work on either of those computers. Somewhere in the middle, I think I helped build a computer that runs a CVS somewhere. But while the applications, I think, for a lot of these things are very interesting, the actual design of the hardware, which is what I worked on, is fairly repetitive and wasn't really too exciting. But the important thing about me working there is when I got bored and decided to quit, I happened to go by the UT campus where Andy Dunn was a professor just starting there, and I happened to see him give a talk, and I think this was the same slide he gave in his talk, talking about imaging techniques to look at stroke and 4-POS stimulation. And I thought, even though I had some sort of background, at least in MRI imaging, but no real background in any sort of medical optics, but I thought this was interesting enough that I wanted to get involved. So I just asked him to join his lab. He said sure, and that's where I spent the next 5 years or so. I guess I should, I was thinking of just leaving this out, but I thought maybe I'll just give a brief background of speckle contrast imaging, even though I realize there are probably a lot of people here that know a lot more about it than I do. But the gist is that you shine diffuse coherent light onto the surface of, in this case, a rat skull, and it creates a static interference pattern. As red blood cells interact with the interference pattern, you get to see this sort of twinkling effect. Anyway, if you can quantify this twinkling and back calculate out the frequency shift, it'll tell you something about the velocity of the particles going underneath, and you can go from the image on the left to the image on the right. The actual math about how that's done is fleshed out in the literature over the years, and it's somewhat complex and requires a lot of estimations. Those familiar with the research probably already know a lot about it, and those that aren't probably don't really care as much. But the process of generating a speckle contrast value and actually relating that just to the intensity fluctuations of the autocovariance of the intensity fluctuations, and then relating that back to the field autocorrelation time, and then that in turn is somewhat related to velocity, is a long process filled with a lot of assumptions. But it's generally well accepted, at least I feel, that for the most part, I'll be talking about these speckle contrast values and saying that they equate at least to relative blood flow. So one of my first experiments, one of the things I wanted to start doing in the lab when I first started was actually trying to adapt this speckle contrast imaging into a neurological microscope that they were using at the nearby hospital. And I think we had an IRB to look at tumor demarcations using blood flow. I think by the time that IRB got approved, I had pretty much graduated. But my colleagues had actually taken this and were able to use it as I was leaving at the local hospital. and they were able to generate speckle contrast images of the human brain. I think it probably will end up having a lot of use. Initially, it was used for tumor demarcation, but I think it has potential to be used for something like judging the efficacy of aneurysm surgeries, and maybe doing so without contrast agents like ICG or things like that. But what I spent most of my time doing was actually working on rat animal models. So I was always asked about how that works if, in terms of, do you remove the skull? Do you thin down the skull? Can it see through the skull? So some people may be very familiar with how it works, but as I always videotape all my experiments, I thought I'd include what we actually do to use speckle on the surface of the rat brain. So we remove the tissue, and we use a lot of dental surgical tools. And here, I think this is a video of me basically thinning down the surface of the skull. I think in this part, I'm just sort of making a hole to inject or to place an electrical probe or something of that nature. But after we thin down the skull, we can put like a well around it and then put some gel to improve surface quality. And that's with the surface intact, you actually get a pretty good view of the surface of the cortex. And then from there, you can actually do speckle contrast imaging on the brain. And this is typically what it looks like. You shine the laser light on the surface, and pretty much in real time, you can generate these speckle contrast images. I think at this point in the video, this was a video I had made for a new journal, a methods journal of visualized experiments that I thought was really cool, at least a cool idea for method papers. And I think I'm just highlighting the ability to sort of select particular regions of interest and then generate in real time plots of blood flow. So I think the experiment I was going to do in the video here was actually prick the surface of the cortex, and then what you should be able to see is an increase in blood flow that we can actually kind of watch happen in real time. And I'll get back to maybe talking about why that's important. I had seen, also one of the first things we'd worked on in the lab was developing the algorithms to look at those changes in real time. I know I saw a paper from some groups here about using GPUs to do sort of the same thing. I mean, as technology advances, I'm sure my phone now can process these images in real time, but it was nice to have at the time, not only to look at slow changes like these depolarizations, which can take on the order of minutes, but even looking at functional changes, which can take on the order of seconds, we can still look at those changes in real time. I included this because I really like this paper. It came out a few years ago. It shows a very useful application of speckle contrast imaging, I thought. So the reason I started looking at these depolarizations is people were interested in looking at them in rats as sort of a corollary to migraines in humans. And one of the hypotheses put forward was that after these depolarizations pass, because of basically you would have ion fluxes that would penetrate from the cortex to the meningeal layers, and then through a long chain of events would affect afferents of the trigeminal nerve. And they wanted to test this out to see if this was true. So they took a speckle contrast image long after the blood flow change had occurred. You can actually clearly see that even though the cortex has decreased blood flow, there's increased blood flow in the dural layers, I suppose you could say. And I thought, well, that actually happened to work out perfectly, because that speckle contrast imaging technique really could help answer at least that part of the vascular hypothesis. But what I was interested in looking at is the part before, the part actually during the actual depolarization, because the consensus at the time, and I suppose still is that when this large change in blood flow occurs, there's no damage to the brain. There's a spreading depolarization, but the blood flow pretty much repolarizes it, and the brain's fine. So in order to test that theory, or at least begin to try to test it, I thought, well, perhaps we could look at oxygenation in the brain. So, of course, the concept of using reflectance imaging to look at changes in oxy and deoxyhemoglobin has been around for a really long time. Absorption spectra of oxy and deoxyhemoglobin are pretty unique, so that if you shine different wavelengths of light on it, you get pretty varied responses. So a lot of this work has been done many years before, but basically using a differential form of the modified Beer-Lambert law and shining light of different wavelengths onto the surface, you can back calculate actual changes in oxy and deoxyhemoglobin. So this was a sort of a diagram of, this is actually a diagram I think I took from Andy of the first system that he had built. And I had made some improvements that I'll probably talk about maybe a little later. But the gist is pretty simple. On one side, you're shining laser light. On the other side, you're shining light, perhaps distributed through a filter wheel at different wavelengths. You have a dichroic and two cameras to basically split the two sources of light, and you can look at simultaneously changes in blood flow and changes in oxy and deoxyhemoglobin. So you end up getting reflectance images like this, the speckle contrast images you've seen before. And the main advantage of being able to, or one advantage of being able to look at these two things at the same time, if you look at a particular blood voxel and you see the blood flow coming in and you see the changes in oxy and deoxyhemoglobin, you can estimate something about the oxygen metabolism. So I ran this experiment during these sort of blood flow increases that happen after the spreading depolarization. And what I found when I went back and analyzed the data is that even though there's a large increase in blood flow, the changes in oxygenated hemoglobin were not very significant. In fact, it was really dominated by deoxygenated hemoglobin, almost implying that the metabolism, the high oxygen metabolism, was creating a situation where supply was not enough to meet demand. And I thought, oh, well, that was really not what I expected to see. I thought it was pretty interesting and pretty repeatable. And I think this was sort of the first paper I tried to write. And I remember the response from the reviewer was basically like, oh, well, this is a good idea because no one's done this before and it really needs to be done. But I don't think those results are right. In fact, you probably made a mistake somewhere because it kind of contradicts what's kind of been out there, at least in terms of point measurements. And after I got over my initial bitterness at that, I decided that he was actually right, and he made a good point. And I really couldn't explain why those results were different. And I thought, well, one way to probably figure out, for a few different reasons, I decided in order to figure out what was really going on, I needed to look at the absolute PO2 values, not look at the relative changes in oxygen and deoxygenated hemoglobin. So I started using a technique based on phosphorescence quenching. A new probe had been developed that basically reacted with oxygen, and it was a phosphorescent probe, so that we inject it into the bloodstream, we excite it, we look at the decay time, and because the probe reacts with oxygen, by looking at the decay time, we can kind of get a feel for the localized oxygen concentration where we excite it. So I went back. Oh, so this is just sort of a description of how the setup was done. One problem with phosphorescence quenching is that it takes a long time, so you need a laser with a relatively low rep rate and a small duty cycle. I did not have that, but luckily we were building a multi-photon microscope in our lab, and we had a very powerful 10-watt green laser. So combining that with an AOM to sort of act as an optical shutter, I was able to create a laser with a low rep rate and a low duty cycle that could basically be controlled digitally, and I could run my experiment and collect my data. And what I ended up doing was I ended up looking at the changes in oxygen during these spreading depolarizations for animal under different baseline conditions and different anesthetics. So initially what I had used was urethane as my anesthetic, which wasn't a popular choice, I had noticed, but it was fairly easy to use, so that's why I used it. But I went back and I noticed that most people that did these results used isoflurane, and they have different channels as an anesthetic. I don't remember off the top of my head, but I think isoflurane's a GABA inhibitor. Well, I won't even say because I probably won't be right. But I recall that the channels that each of these anesthetics use are different. And what I looked and what I found is that the baseline PO2 is actually largely decreased in these urethane anesthetized animals to the point so that they were almost hypoxic, which could most likely explain the results I was seeing, because the isoflurane hypoxic animals actually had higher baseline PO2s than the urethane anesthetized animals. And when the animal was hypoxic or under urethane, I did not see a large increase in oxygen value, even though I always saw large increases in blood flow. So I went back, sort of repeated this protocol with the setup,
and basically saw that my oxygenated hemoglobin data basically matched my PO2 data. And if I had the anesthetic and the animal had a normal baseline, I would see normal increases in oxygenated hemoglobin and significant increases in absolute PO2. When the baseline was low, I did not see those changes. So I guess I like that as an example of basically putting together more information to answer sort of an interesting hypothesis. As an aside, one of the other colleagues in my lab that was building the multiphoton imaging setup was also using the probe to look at PO2 values at different lengths, at different points along the same vessel in hypoxic and normoxic animals, or normoxic and hyperoxic. And it was just another interesting case of, I think he's one of the first people to use the two-photon setup to actually map oxygen along the same vessel. So one problem I had that he did not have is, because the multiphoton setup inherently has a scanning laser source, is he had this spatial resolution, whereas I was shining light over an entire generic tissue source. So I really had no spatial resolution. I was using a PMT, and even though I could gauge sort of the general baseline PO2 levels, I really had no idea what was going on in terms of what was happening in particular areas. And around the same time, up the street, Texas Instruments they were developing their light projection televisions. They had those commercials with the elephant that I didn't really understand. But they wanted to use these chips for medical applications, and they basically gave us one. And it's a fairly advanced spatial light modulator. And one application that seemed almost obvious is, well, if I run my light source through, or basically reflect it off the spatial light modulator, I can use the high resolution of basically this television to select particular regions of interest. And I could co-register it with the speckle contrast image. So while I may not have point-by-point pixel resolution of PO2 changes, I can sort of pick out the regions of interest that I care about and look for the PO2 values in only those particular regions. So I tested this out in stroke models, because I started looking more at strokes. This is kind of a stroke technique where you see the before and after. Blood flow obviously decreases. But what's more interesting is that I can use the DMD, the spatial light modulator, to highlight particular veins or branches of arteries or the parenchyma, and then I could watch those PO2 changes. I could actually watch those PO2 changes during while the stroke was occurring. So I think in this movie, right now I'm about to apply this topical vasoconstrictor that decreases blood flow. And you can see as blood flow decreases, I can actually look at changes in PO2. And I guess one of the interesting things as well, just the fact that it can be done, but you can see, in addition to mapping out blood flow changes in the vessels, while you'd sort of expect that the arteries would be the most, or suffer the most from this constriction, they'd suffer the most loss of blood flow. It's actually the parenchyma that's surrounding these blood flow or surrounding these arteries that's suffering the most significant changes in PO2 loss. So that's just this again. And then just another example, we can induce a stroke, and if you're not interested in particular vessels, you can sort of use it to map out the penumbra on the core, basically looking at different distances from the stroke induction site, and kind of mapping the PO2 values along different regions. And then sort of by accident one day, what I found out you could do is I could actually induce strokes. I could use the spatial light modulator to highlight a particular region in a vessel, and then I can induce a photocoagulant and turn the DMD on high power, excite the coagulant, and actually just occlude a particularly small vessel, and then kind of measure the PO2 around that vessel and see what's going on. So in this movie, I turned on the DMD to occlude that vessel, and the map on the right kind of shows the blood flow changes in real time. And I suppose what I envision is happening is that as blood flow decreases to that small area, what you'll actually see is that it induces a spreading depolarization, very similar to what happens when you prick the surface of the brain. The brain responds by sending a large increase in blood flow to the area to sort of try and re-energize the cells that are depolarized. But essentially, it fails because the occlusion is still there, And actually what you see is that after the depolarization, it seems to waste so much energy that the surrounding tissues actually decreases in blood flow altogether. So I thought that was interesting, just kind of an interesting application sort of to answer the question, well, why not just use a scanning source to look at these different regions? And for something like occluding a small vessel, you're not really interested, you're really almost interested in sort of radial projections, collecting data at different distances from wherever that occlusion is. And that the DMD is actually a good way to do it because it can create very arbitrary sized shapes. And in addition to that, you get the advantage of the signal-to-noise ratio of basically exciting that entire shape as opposed to scanning pixel by pixel and only exciting that small region. So one of the last things I tried to do was basically combine everything together. It's not too difficult. It's just really a lot of bookkeeping, but combining all those three results and then kind of teasing out all the data to be able to look at all the data at the same time. Luckily, all the excitation and emission spectra we're using were all nicely separated, so I was able to, with the right dichroics and right filters, I was able to kind of build something to look at all these things at the same time. Just shows an example of the separation that's going on, and basically it worked. We could look at all those parameters at the same time instead of having to look at them individually. And even though I felt I was pretty much done with my PhD at this work, there was one general problem, is that for all practical purposes, the system was, it was very expensive, and it was basically impossible to use. I mean, I could use it because I had spent, a year of my life putting it together, but practically speaking, I mean, I remember I was borrowing the laser source for the phosphorescence quenching from the 2-photon setup that sat in another lab, so I ran a fiber through the roof to actually deliver that light, and there was something else in another room, and each system was run by a separate computer, so starting it involved running around to about 5 different places and hitting a bunch of different buttons, and collecting or at least analyzing the data was almost orders of magnitude worse. And realistically, I knew as soon as I left, this project would basically be vultured for scrap parts and never be used again. Even though the results were relatively interesting, if no one can use it, it's kind of worthless, at least in my mind, because I'm always thinking about, I'm sort of used to this industry perspective of like, well, will anyone buy this? Will there be a customer for this product? And for this product, the answer was clearly no. So I made a lot of changes to the system. The general theme of the changes was going from analog to digital, replacing a lot of the light sources with LEDs that could be digitally controlled, replacing photomultiplier tubes with APDs, using more efficient cameras, moving away from cooled 16-bit when I could get the same information from 12-bit cameras. And in addition to making the system a lot cheaper, which was nice, but since I don't really pay for it, I don't know that I cared about it being that cheap. What I thought was nice is because everything was digitally controlled, I could pretty much run everything from 1 or 2 computers. And really, it got to the point where you could just hit a button and all the excitation sources would turn on. There was no turning on a lamp, turning on a laser in another room. Everything could be digitally controlled. And also because it could be digitally controlled, it made it a lot easier to analyze. Because I had all these digital signals as cues, I know exactly when each thing started and stopped. And it really became a lot more usable. I think it would be a lot more practical to use the system today. And that's actually what it looks like. So those are all the components sort of hidden in there. I think I made a movie of how it worked. That's the sample rat. On its back is, I think, a cuvette of some phosphorescent dye. So it's sort of a track lighting setup stage where all the LEDs are kind of firing. Those are the camera images from the reflectance imaging camera. I obviously used this movie more than once. That's the same movie you saw before, but I just used it as a demonstration for showing the blood flow changes that you can see. I think the next thing that I focus on is the spatial light modulator. And you can kind of see how you will see in a second how it works with the basically different shapes being highlighted. At this, it's kind of like a triangle and a square. Obviously, it can do much more complex shapes, and it can do this at several thousand hertz, because it's a television. It needs to do this at hertz to develop good contrast. And that's sort of using a scope, you just kind of see the decay, the phosphorescence decay. So that's pretty much how the system looks and works. There's an example of using the results to look at functional activation. There's sort of the stimulus at the bottom, the somatosensory cortex region of the cortex kind of in the top left. So when you make the measurements in the top left, you see the increase in hemoglobin. Far away, you see kind of a small residual increase in blood flow, but no real change. So it was nice in that it worked. And I guess my overall conclusion from all this was that twofold. One is that it's nice to have all this information. It paints a good picture when you're looking at trying to figure out what's going on in a physiological phenomena. The more data you have, the better, at least in terms of if you're trying to really understand what's happening. And two, it's important to make, if you design instruments like I do, it's important to actually make it so that it's practically useful, so that hopefully maybe someone else may be able to use it to look at another physiological phenomena that's of interest to them, because obviously not everyone's interested in the same thing that you're interested in. I know it's always kind of iffy when someone talks for a while about one thing and then says, all right, well, now I want to spend another hour talking about my next area of research. So I'll do that, but I guarantee it's very quick, and I think it's very interesting, at least to me, which is basically how I got started working in my postdoctoral life, which is essentially, to summarize it, some people had seen some of this, and they thought, well, this is interesting, and I really like it. But this brain stuff is, while great, I actually work a lot with ophthalmologists. And I think a lot of these tools could be used very impressively to perhaps maybe do some early detection of diabetic retinopathy. In Texas, diabetes is a huge problem, and they estimate that a significant proportion of the elderly will start to basically go blind because of the huge diabetes problem. So we got a lot of funding to look at this, and one of these professors asked me, well, can you just point this thing in the eye and give us the same kind of information? And I thought, yeah, sure. I mean, I didn't see why not. I mean, tissue is tissue, and I really built a device to look at tissue. And if anything, I mean, retinal tissue is very similar to brain tissue. So I didn't really perceive any problems. So I took the system there. I tried it, and this was my first image. That's actually better than I remember that image being. But it was pretty terrible, obviously. So then I worked on it a few weeks, I think, and it became slightly less terrible, but still quite terrible. And I think on the left is a speckle contrast image. On the right is the reflectance. You can sort of make out what may be a retina, what I would hope is a retina, but clearly nothing good enough to analyze. So I obviously realized I had a few different problems. One is getting light into the eye is kind of tricky. You obviously can't stare into someone's eye and see their retina like you can when you make an optical window to the brain. And two, the eye is very curved. The brain is not. So I started reading papers of people that actually do this more diligently. And I noticed one common technique is to use a contact lens. So using a contact lens to correct for the curvature of the eye, and then also basically changing the path length of the excitation light to get better results. And this was from another paper that someone did, and contact lens technique was pretty common. So I basically copied it, and I got somewhat better images, still not great. And while they were good enough to do some kind of analysis, at least separating out retinal vasculature from the choroidal vasculature, it was actually a pain to set up. I mean, not too difficult, but certainly too difficult that I couldn't give it to somebody else that wasn't familiar with working with these kinds of imaging systems and say, hey, take some images of the eye. It was, putting the contact lens on and aligning everything was just too difficult. It was a pain for me to do it, and it was definitely going to be a pain for someone else to do it. So I had sort of had the same experience of if I leave the system like this, no one will ever use it. So in looking around, I ended up using an otoscope, which is basically a sort of endoscope that an ear doctor will use to take a look into your ear. And it actually helped me solve both problems in a very simple way, because the light source for the otoscope actually travels through the viewing, through the viewing port, and also the the optics, in order to develop sort of a wide field of view, was actually well designed to handle the curvature of the eye. And the beauty of the system is, one, it was very cheap, and because there were other systems that did a good job of solving this problem, unfortunately they cost, hundreds of thousands of dollars. And I think this was very effective and also very cheap. But more importantly, it's very easy to use. The system's almost handheld, and you really just kind of hold it up to the animal's eye, and that's it, and hit the button and take the image. So I did that, and I was able to get what I thought were pretty good images. And I've been doing a lot of work trying to basically characterize the response to different things like gas challenges and functional responses, and going through and doing that in healthy animals, and then hopefully with the long-term goal, then going back and look at diabetic animals, because there's a well-established animal model for diabetic retinopathy, and once you characterize the baseline, maybe going back and seeing, well, what can we do to predict sort of these changes earlier on. And that's pretty much it for me. So these are the people in my new and old lab, and that's where I'm told the money comes from for me to work. And I'll take questions if people have.